In recent years, a deadly virus has emerged, striking fear into communities worldwide. Today, we delve into the depths of this lethal pathogen. Welcome to our YouTube video on Nipah virus, the deadly threat. Let's get started. Brief history of Nipah virus. The Nipah virus has a relatively short but impactful history since its discovery in the late 1990s. Here's a brief overview. The Nipah virus was first identified during an outbreak in Malaysia and Singapore in 1998. The outbreak initially affected pig farmers, who experienced severe respiratory and neurological symptoms, leading to a high fatality rate. The virus was traced back to fruit bats, which served as the natural reservoir of the virus. After the initial outbreak, Nipah virus resurfaced in Bangladesh in 2001, causing sporadic cases and outbreaks primarily in rural areas. Subsequent outbreaks occurred in Bangladesh in 2003, 2004, 2005, 2007, and 2008, with varying levels of severity and mortality rates. In 2018, a major outbreak occurred in the state of Kerala, India, resulting in numerous fatalities and significant public health concerns. Scientists and researchers have been studying the Nipah virus to better understand its transmission, pathogenesis, and potential treatments. The Nipah virus continues to be a concerning global health threat, and ongoing research and surveillance efforts are essential to mitigate its impact and prevent future outbreaks. Structure of Nipah virus. The Nipah virus belongs to the Paramyxoviridae family. The Nipah virus is enveloped, meaning it has an outer lipid membrane derived from the host cell during the viral budding process. The Nipah virus has two major glycoproteins present on its envelope: the attachment glycoprotein, G, and the fusion glycoprotein, F, that are involved in viral attachment and fusion with host cell membranes. Beneath the viral envelope, the Nipah virus possesses a helical nucleocapsid structure. The nucleocapsid is composed of the viral nucleoprotein, N, phosphoprotein, P, and large polymerase protein, L. The Nipah virus has a non-segmented, single-stranded RNA genome. The viral genome is negative sense. The genome encodes several structural and non-structural proteins necessary for viral replication, assembly, and interaction with host cells. How Nipah virus transmits. The Nipah virus can transmit from animals to humans, as well as from person to person. Here are the primary modes of transmission. Direct contact with infected bats. Fruit bats of the Pteropodidae family, commonly known as flying foxes, are the natural reservoir hosts of the Nipah virus. Direct contact with bat saliva, urine, or droppings can lead to transmission. This can occur during activities such as handling bats, consuming fruits contaminated by bat droppings, or being near to bat habitats. Consumption of contaminated food. Nipah virus can spread through the consumption of fruits that have been contaminated by bat saliva or urine. When fruit bats feed on fruits and deposit saliva or urine on them, the virus can be present on the fruit's surface. If humans consume these contaminated fruits without proper washing or peeling, they can become infected. Close contact with infected animals. Nipah virus can be transmitted from infected animals, particularly pigs, to humans. Close contact with infected pigs, their bodily fluids, or tissues can result in transmission. Human-to-human transmission. Nipah virus can also spread from person to person, primarily in close contact situations. Respiratory droplets generated through coughing or sneezing by an infected individual can lead to transmission. Pathogenicity of Nipah virus. The Nipah virus, an emerging zoonotic pathogen, is known for its high pathogenicity and ability to cause severe illness in humans. Here's an overview regarding the pathogenicity of the Nipah virus. After exposure to the virus, it primarily targets the respiratory and central nervous systems. In the respiratory system, it can cause severe respiratory distress and pneumonia. In the central nervous system, the virus leads to inflammation of the brain, resulting in neurological symptoms and potential long-term complications. The host immune response plays a crucial role in the pathogenesis of Nipah virus infection. The virus can evade and suppress the immune system, allowing for uncontrolled viral replication. Excessive immune activation and cytokine release contribute to the development of severe disease, including encephalitis and multi-organ failure. Nipah virus infections can be severe, with high mortality rates. The fatality rate has varied, ranging from approximately 40% to over 90%. The pathogenicity of the Nipah virus poses significant challenges for public health and requires vigilant surveillance and outbreak response efforts. What are the symptoms of Nipah virus infection? The symptoms of Nipah virus can range from mild to severe and typically appear within 4 to 14 days after exposure to the virus. Here's a brief overview of the symptoms. Fever. Nipah virus infection often begins with a sudden onset of high-grade fever, typically above 100.4 degrees Fahrenheit. Headache. Headache is a common symptom and can range from mild to severe. Respiratory symptoms. Respiratory symptoms may include cough, sore throat, and difficulty breathing. In severe cases, respiratory distress and pneumonia can occur. Neurological symptoms. Neurological symptoms are prominent in Nipah virus infection and can include dizziness, confusion, disorientation, and altered mental status. Encephalitis, inflammation of the brain, can lead to seizures, coma, and potential long-term neurological complications. Muscle pain. Myalgia or muscle pain is a common symptom associated with Nipah virus infection. Fatigue. Fatigue and weakness may be present, contributing to a general feeling of malaise. Gastrointestinal symptoms. Some individuals may experience nausea, vomiting, and abdominal pain. Diarrhea can also occur in some cases. It's important to note that the severity of symptoms can vary among individuals, with some experiencing mild illness while others develop severe complications. Severe cases of Nipah virus infection can lead to respiratory failure, encephalitis, with high fatality rate. If you suspect Nipah virus infection, it is crucial to seek immediate medical attention. Early diagnosis and appropriate medical care are essential in managing the disease and preventing its spread. Diagnosis of Nipah virus infection. Here are the primary methods used to diagnose Nipah virus infection, which include clinical evaluation. A thorough assessment of symptoms and medical history can help in identifying potential Nipah virus infection. The presence of characteristic symptoms such as fever, headache, respiratory distress, and neurological symptoms can raise suspicion. RT-PCR test. RT-PCR is the most commonly used to detect the presence of Nipah virus genetic material, RNA, in various body fluids, blood, respiratory secretions, cerebrospinal fluid. These tests is typically used during the early stages of the infection. Antibody detection. ELISA and other serological tests can detect antibodies specific to Nipah virus in blood samples. These tests are typically used during the later stages of the infection. Epidemiological investigation. Gathering information about potential exposure to Nipah virus is crucial in the diagnostic process. This includes evaluating travel history to regions with known Nipah virus outbreaks or exposure to animals that could carry the virus, particularly fruit bats and pigs. What is the treatment for Nipah virus infection? Currently, there is no specific antiviral treatment available for Nipah virus infection. However, supportive care and symptom management are essential in providing relief to affected individuals, which include hospitalization. Individuals with suspected or confirmed Nipah virus infection should be hospitalized in isolation units. Regular monitoring of vital signs, including temperature, blood pressure, heart rate, and oxygen levels, is important to assess the patient's condition. Maintaining proper hydration and electrolyte balance is crucial, especially for patients with respiratory distress, vomiting, or diarrhea. Patients with severe respiratory symptoms may require supplemental oxygen or mechanical ventilation to support breathing. Prevention of complications. If seizures occur, appropriate anticonvulsant medications may be administered to control them. Medications may also be prescribed to reduce fever, headache, or nausea. Experimental therapies. In some cases, experimental antiviral drugs or therapies may be considered on a case-by-case basis under strict ethical guidelines and in consultation with experts and regulatory authorities. How to prevent Nipah virus infection. Here are important preventive measures that can help prevent Nipah virus infection. Avoid direct contact with bats. Avoid direct contact with bats, especially fruit bats or flying foxes, which are known to carry and transmit the Nipah virus. Do not handle sick or dead bats, and avoid consuming bat meat or products derived from bats. Practice good hygiene. Wash hands frequently with soap and water for at least 20 seconds, especially after being in contact with animals or animal products. Food safety measures. Avoid consumption of fruits that may have been contaminated by bat droppings or saliva. Ensure that fruits are properly washed and peeled before consumption. Animal husbandry practices. Implement appropriate biosecurity measures in pig farms to prevent transmission from bats to pigs, as pigs can act as intermediate hosts and transmit the virus to humans. Implement infection control measures. In healthcare settings, adhere to strict infection control practices, including proper use of personal protective equipment, PPE, when handling suspected or confirmed Nipah virus cases. Public health surveillance and response. Establish robust surveillance systems to detect and monitor Nipah virus outbreaks. Promptly report any suspected cases to public health authorities for timely investigation and response. Public awareness and education. Educate the public, healthcare workers, and individuals at risk about the transmission routes, symptoms, and preventive measures of Nipah virus infection. Promote awareness about safe food practices and the importance of avoiding direct contact with bats. Conclusion. Remember, knowledge is power, and staying informed is crucial in combating infectious diseases like Nipah virus. If you found this video helpful, give it a thumbs up and don't forget to subscribe to our channel for more informative content. Thanks for joining us. Stay safe, stay informed, and together, we can fight against Nipah virus.